Okay, well, thank you to the Society for asking me to speak. There's been a bit of a trailer about some of the things that I'm going to talk about, and that's all very useful. But I do want to emphasize what Andy just said, that your donations of samples to laboratories for research, which is mutually beneficial, is absolutely essential. Without that, we can do nothing. So it's a partnership, this kind of research. And I just want to tell you something about the history of what's gone on in CLL. Again, some of this has been talked about. This is off the internet, so we've all got that particular picture of CLL cells in the blood with red blood cells in the background. And the incidence figures have been talked about too, that it is a disease of older age group, but also that it's a very common disease compared with acute myeloid and chronic myeloid leukemia. It is much more prevalent than the other leukemias. So it has to be taken very seriously. This has also been talked about to some degree, that the cells from which CLL develop, and CLL cells themselves, are virtually indistinguishable. So when you look down the microscope or look at a picture of the cell as this is, you see the various components of the cell. The nucleus, which John Strefford spoke about, that's the information center of the cell, controls everything the cell does via its DNA content. The cytoplasm, where many of the functional molecules sit, and then the surface membrane, where molecules sit which talk to each other, so cell-to-cell communication, cell-to-microenvironment, which are very important in maintaining the cell viability. Now, CLL cells look remarkably like normal blood lymphocytes, and CLL is an indolent disease. It's not like acute leukemias where the cells are very large, dividing very rapidly, very dangerous, and have to be treated immediately with chemotherapy before we can even study them. Chronic lymphocytic leukemia is an indolent disease. And we have to change our thinking on cancer. Although it is cancer, it is an indolent cancer. So we have to think very carefully about whether it should be treated, watched and waited, etc. So the questions that arise are, what is the cell of origin? This turns out to be very important. How does a normal B cell become a CLL cell? What was its parental cell? What is the key transforming event, if there is one? Why does it become a CLL cell? Why do CLL cells divide and survive? Why don't they just die off, as many normal cells do? Why does the rate of disease progression vary so widely? This is something that's very puzzling to patients. I think a number of patients go into the doctor for other reasons. They're diagnosed with CLL, and then they don't know whether it's going to be rapidly progressive or just not progressive. These kind of questions are essential for the patients to know about. Now we can say with a fair degree of certainty which category you might be in. If the category is an aggressive tumor, it can be treated more effectively. If it's not, it can be left alone. So which patient should be treated and which treatment is useful? And just to go back over a bit of history, I think Sian mentioned that normal lymphocytes are composed of two sets of cells, B and T cells. And this is because the immune system has to do several things. It has to fight virus infections. And viruses are very clever at getting inside cells and avoiding everything. They have to be attacked by T cells, and that's what's called cellular immunity. The other side of immunity is an antibody-mediated immunity. You all know we've got antibodies in our serum circulating all the time, as Sian said, looking for pathogens and foreign antigens. So this is antibody and deals with mostly with bacterial infections. This is a generalization, but that's the way the immune system's split up. Max Cooper, whom I met in Sydney in September, he just had his 70th birthday party, discovered that the cells which we saw circulating in our body were in fact composed of these two subsets of cells, B and T cells. Does that matter for CLL? Yes, it does, because CLL comes from B cells. There are tumours of T cells. In fact, there's a very small minority of CLL which is from T cells. It's such a small minority, it's not worth worrying about. The vast majority of CLL cells comes from B cells. And this, which you don't need to read in detail at all, is the history of the discovery of B cells and what they do. And what it does is illustrate to you the immense amount of work and research that goes into these kind of studies. It started in 1890. I won't go through all the steps, but the steps proceeded along, and this is only up to 1988, and it still goes on now. On its way, some important findings came up. This is the one Max Cooper found about the separation of B and T cells in 1965. And these are just a few of the Nobel Prizes that have been won during this trajectory. So this has been important and difficult work to get to where we are now. This is a very early paper from a journal called Blood, on which I'm associate editor. It's based in the United States. It's the major hematological journal that we have in the world. And this was a very early paper in 1967 by a very famous hematologist, an American hematologist called Damashek, and he was looking at CLL. So no color photography there. He was just looking down the microscope, looking at these B lymphocytes, and he described it as an accumulative disease. In other words, the cells keep growing, developing, dividing very slowly of immunologically incompetent lymphocytes. That means, although they're B cells, they're not going on to produce antibody or anything useful. They're just incompetent. He didn't then know how to recognize B lymphocytes. That had just been described, and there were no reagents that could be used to say that these were B cells. So that's as far as he got then. The next step relied on these two guys, who are Caesar Milstein and George Kohler. They were in Cambridge, and they were looking at something completely different, but they developed monoclonal antibodies. They developed the technology whereby you get one antibody from one cell, and that antibody would be very specific for what you decided to direct it against. These became reagents for describing cells, and this is a CLL cell, and these CD molecules are all defined by monoclonal antibodies that have been raised, again, in the way that Cian described. So it became clear that CLL was a bit unusual as compared to normal circulating B cells in its so-called phenotype. These monoclonal antibodies were used to look at the phenotype. And what they display is CD5, CD23, and like all B cells, an immunoglobulin molecule on the surface. So CD5 and CD23 were unusual. And that was a diagnostic, and still is, diagnostic marker. You've probably got it all on your information at the hospital or for yourself. These are CD5 positive, CD23 positive, low surface immunoglobulin, and that's part of the diagnosis. There are lots of other molecules too, and their functions are to interact with the microenvironment, to accept growth factors, etc. We won't go into any of that now, but these two guys got the Nobel Prize in 1984 for raising these monoclonal antibodies, which are now in use as diagnostic markers and as treatment. So they have distinctive cell surface markers. But when you look at a real B cell, this is how they look. They're crawling all over the surface, looking for molecules. All those molecules that I just showed defined by monoclonal antibodies are on these projections. The cell is looking out for other cells, or something to stick onto, how to stick down and divide, etc. So this is a more realistic view of a B cell, and that's a normal B cell. So the next step that was needed was to look inside the cell, and John Strefford has talked about the genetic components of the cell. In order to do this, we needed another discovery, which was made by these two guys, Francis Crick and James Watson. You've all heard of the double helix and read about the discovery of DNA. This is the DNA chalked on the blackboard by James Watson. Essentially, it's a double helix in which bases are connecting the two strands of the helix. And it immediately showed that these two strands could come apart and replicate. So this specific pairing suggests a possible copying mechanism for the genetic material. This was a seminal finding. We now know how cells can divide and take all their information with them using DNA replication. Nobel Prize in 1962 for that. But this guy was equally important because he worked out how to sequence the bases in the DNA. So he cracked the code. DNA carries a code for everything that it does. So the bases, the 4 bases, are in sequence, and each base makes, the triplet base makes an amino acid. So he had to crack the code. He did it first for proteins, got the Nobel Prize in 1958, and then he did it for DNA in 1980. So he got 2 Nobel Prizes in the same overall subject. So without these guys, we wouldn't be talking in the way that we do now. The genetic revolution followed from these findings and immediately became relevant to cancer. It increased our understanding of the disease process. It offered new rational treatment options, targeting certain genes which we now knew were changed or overexpressed. And it continues to allow tailoring of treatment. We've talked a little bit about this. Each one of you with CLL is an individual, and your CLL will be different from the other person's CLL in possibly minor or in sometimes major ways. Tailoring of treatment is very important for the patient. We don't do overtreat or undertreat. So now we come on to this molecule, the immunoglobulin. Francesco asked me to talk to you about the unmutated and mutated aspects of immunoglobulin. And this I will try and take you through without overwhelming you. The first thing to remember is that in CLL, many cases have few or no chromosomal changes, apart from deletions at 13q14, which upregulate BCL2. You don't need to know anything about that, but it keeps the cells alive. So many CLL cells don't have many other genetic changes. They look so much like normal cells. In some respects, they behave like normal cells. What they do is exploit the fact that they can live longer. So maybe Damashek was right. These are long-lived cells, and they can exploit the environment. So here we have an immunoglobulin molecule, which is the receptor on the surface of these CLL cells. This is the critical receptor for all B cells and for CLL cells. Its normal function is to sense the environment for antigen, for foreign pathogens. What does it do in CLL? It sees something else. The analysis of this immunoglobulin on CLL cells provides insight into how the tumor develops, and it provides a prognostic factor and a target for new drugs. So it's a very important molecule to understand. Here it is drawn in a slightly different way. It's on the cell surface. This is the surface of the cell. This is the immunoglobulin molecule. These are co-molecules which allow signaling of this immunoglobulin's activity into the cell. You don't want to know what goes on inside the cell. It is extremely complicated. Many phosphorylation steps and various other changes go on within the cell which lead to all sorts of functional changes which allow the cell to proliferate and survive. All you need to know about for this argument is that the cell proliferates and divides after it's received signals through its surface immunoglobulin. The cell division occurs and differentiation to antibody-producing plasma cells. In a normal B cell, an antigen will come in. I've shown a pathogen here. This is actually Helicobacter pylori, which you might have heard about in association with gastric lymphoma. The cell senses that pathogen or antigen, signals down and starts cell division and differentiation to antibody-producing plasma cells. So the antibody is then produced, which will fight the organism. It's a very nicely organized system. Now, how does it do it? Because in normal B cells, the question that arose was, how do normal B cells cope with all the different pathogens that are being thrown at us? I mentioned Helicobacter, but think of all the viruses and all the bacteria that we have to deal with every day. How does the immunoglobulin molecule itself deal with all these different ones? What it does is that there's a very clever rearrangement process, which means that every B cell has a different sequence in these red areas. And it was DNA sequencing that allowed us to see this. So each molecule can react with just a limited number of pathogens, but overall, we can cope with all of them. So the process by which it does that is a magical process of genetic modification of these so-called variable regions. Now, this is where it gets a bit hard because there are two processes. One is a rearrangement process called a VDJ recombination, where three genes join together in different ways to make different sequences. The other is easier to understand, somatic hypermutation, which means there are changes in those bases that Crick and Watson described, which change the sequence of the protein. So it's a way of generating diversity. It was called God when it was discovered, a generation of diversity, whereby our immune system can see everything that's out there. This is important for CLL, and I'll explain why. This is normal B cell development. You start in the bone marrow, here with a stem cell, and you end up with an antibody-forming plasma cell. The B cell has to undergo all these changes to get there. And it does so by this VDJ recombination, which I mentioned, and by the somatic mutation, which goes on here when it's seen antigen. The somatic mutation process was another Nobel Prize, actually part of Cesar Milstein's Nobel Prize. He described how an antibody variable region, the red bits, start off with a certain sequence, but then accumulate all these mutations as the antibody develops. It's a way of an antibody getting better and better at seeing the antigen or the pathogen. So this is a change in DNA structure, sequence, in the variable regions. And it's a huge rate, 1 million times the background rate of mutation. And these are natural mutations. But they are quite dangerous because we're changing DNA sequence. And the price we pay for producing these high affinity antibodies is the development of tumors, because DNA is being moved around in the VDJ recombination and in somatic hypermutation. As soon as you expose DNA to mutational change, there are dangers. And the price we pay is the development of all the B-cell tumors, some of which you'll know. You certainly know this one. But acute leukemias develop at this point in early B cells. All the non-Hodgkin's lymphomas develop during somatic hypermutation, and multiple myeloma develops later during a further process. So the price we pay is the susceptibility to some of these B-cell tumors, all arising at different points. Now, the point about CLL is that it rises at 2 points. That's very strange. For many years, people thought it was a single disease. But no, it arises before the activation of somatic mutation and after the activation of somatic mutation. This is why we have unmutated and mutated cases. And this matters. So what you get, these cells that often look the same from patient to patient, actually consist of 2 diseases. One from these carrying unmutated regions, these B cells before somatic mutation, one with mutated V genes arising after the activation of somatic mutation. Now, the proportions in the whole population are about 40% of patients have this one, and about 60% have that one. But that percentage varies according to whether you're a tertiary referral centre or whether you're Bournemouth, where everybody comes in off the street. But this is the kind of proportion that we found. And this was our finding here, that not only is this interesting biologically, but it's vital in terms of pathogenesis and the tumor behavior, because look at this. These are the cases with the mutated V genes, and this is survival, and this is our paper in 1999, which is a historic paper, and these are the ones with unmutated V genes. So the survival curves separated out. We were absolutely shocked when we saw this, to see such different tumor behavior. And it accounts for the fact that there's such heterogeneity in the way patients go. Which category are they in? It's not absolute, but it's a very good prognostic indicator. It remains the best prognostic indicator. Even after people have tried to find other substitutes that go with it, it remains the best prognostic. In fact, there are a couple of exceptions, but it's pretty minor. It's been verified by other people. This was a verification that came 5 years later from another study. And we shared this finding with an American group. So it was a Southampton finding and a New York finding, which we published together. This was important within the CLL community. So the Binet-Rye medal, which is using that classification system, these two guys, Binet and Rye, developed the first classification system for CLL. Terry Hamlin was awarded the Binet-Rye medal in 2003. He was my partner in the paper. I had to wait 12 years before I got mine, but I was the first female and the first non-clinical scientist to get it, so it was worth waiting for. And this was the presentation, which occurred in 2015. This is actually Kanti Rai, who was responsible for the Binet-Rye classification. This is Michael Keating, who works at the MD Anderson Hospital in Houston, in Texas, who's a very important name in the field of CLL. So, what's going on then? We've got two tumors, really. They're both activating these pathways. So there's something which is stimulating this surface immunoglobulin of CLL. We don't know what it is. It's something that lives in our own bodies, what we call an autoantigen. So you know about autoimmune diseases, there are many of those. Some autoantigen is stimulating both subsets of CLL. The stimulation occurs in the lymph node and to some extent in the bone marrow. This is Ki67, which is a molecule, an antibody again, that sees dividing cells. So cells divide in the lymphoid tissue, and you can see a rather poor outline here of what's called a proliferation center. This is what's going on in the lymph node of patients with CLL. The cells divide in these rather loose aggregates called proliferation centers. They do not divide in the blood. They're just going around in the blood. They have to go through the tissue, settle down, see this, whatever it is, and we're still not sure what it is. Let's call it an autoantigen, and this will fire the proliferative events. You can see it doesn't happen in the peripheral blood. It only happens in the lymph node, where the cell division is occurring. Now, Francesco has spoken about the consequences of these findings. The fact that this signaling pathway is so important in CLL immediately alerted the drug companies to ways of interrupting it, specifically. So this shows one of the components, so-called BTK, Bruton's tyrosine kinase, which was the first target for a drug. So they're going for this pathway. And the first trials that came out were very interesting, and Francesco's described them to some extent. The patients start off with a count which is fairly significant, and it starts to come down, but initially it goes up, and then it comes down. The reason it goes up is that the cells, and it scared the people doing the trial to death when that started, because you can imagine the patients have a count of 50,000, and then go up to 100,000. Crikey. We've stimulated the tumor, but they hadn't. They'd relocated the tumor from the lymphoid tissue into the blood. So what happens is the cells are dragged out of the lymphoid tissue, and they notice the lymph nodes went down, and they go out into the blood, and then they gradually peel away. And the patients go into complete response, partial response. One mustn't overstate this. Upfront single trial patients, 30% failed to respond, another 30% discontinued because of adverse events. I don't want to talk it up too much, and I'm not a clinician. You'll hear better descriptions from Francesco. But it no doubt revolutionized the whole approach to CLL. Somebody asked a question, a very good question, about whether unmutated and mutated CLL respond in the same way. Well, the answer is they don't seem to. Although there are fewer mutated CLL patients treated because their prognosis is better, they're probably left without treatment. In terms of ibrutinib, the unmutated seem to respond better than the mutated. These are the mutated ones. This is a kind of diagram you may not be familiar with, but take it from me that it means that the unmutated respond quite well, but the mutated don't respond quite so well to ibrutinib. So there are differences. And this is summarized in perhaps a more digestible form here, where this is showing patients going into complete response. This is a paper from Ohio in the States, showing the mutated, again, don't respond particularly well, but 6 out of 5 patients went into complete remission on ibrutinib if they were unmutated. So it looks as though there are differences in the way the two subsets respond to therapy, not unexpected. And I think this was shown by Francesco, showing chemoimmunotherapy, fludarabine, cyclophosphamide, and rituximab on the two subsets of CLL. And one of the exciting things is that although this is a very fierce therapy for people with mutated CLL, it does seem to drive some of them into what could be called a cure. And the clinicians are very excited about this plateau, as Francesco described. When you get the plateau, that might mean the disease is gone completely. So there may be a cure coming up. Watch this space. It takes time for all this to be worked out. So, in summary then, and I don't want to bore you with any more, the two subsets of CLL which we described and are clearly there have to be taken seriously. There is some overlap, but there's no doubt these two tend to behave differently. This is the survival curve again. They all express this surface immunoglobulin. They're all encountering something, autoantigen, in the body. We don't know. These are derived from cells before somatic hypermutation. Those are derived from the more mature cells after the activation of somatic mutation. There's a balance here. In summary as to what they're doing, these cells are proliferating more. Their receptor is more active. There is less anergy. Anergy means that when the cell encounters antigen, it may or may not respond by proliferation. Sometimes they just go to sleep. And these cells tend to go to sleep more easily when they encounter this autoantigen rather than going into proliferation, which is more likely in this subset. So it's a differential. It's not totally different. There's just more proliferation and less energy here, less proliferation and more energy there. However, if some of you have got either of these, don't immediately think, I'm in that category. Well, think you're in that category, but be careful because there's a lot of heterogeneity, particularly in this mutated subset. Doesn't mean to say it's not going to give you trouble. It might not, but there is heterogeneity, and the clinicians have to look at the whole picture of the disease before pronouncing that it doesn't matter, you don't have to come anymore to the clinic. But these are less susceptible, initially at least, to those drugs aimed at this pathway which I've mentioned. These are more susceptible to these drugs. So these are the people who've done the work. Francesco came some years ago to do a research degree in my lab, and it was a great pleasure to have him there. Then he went off back to Siena, the most beautiful city in the world, where he trained in hematology, and then for some mysterious reason, he's come back as a fully fledged hematologist with all the scientific training that I was able to give and others in the lab. So it's very good to have him back running this aspect of the hematology clinic and working with Sian and others as well. Graham Packham is a scientist who's very interested in the signaling pathways that are activated within cells. So we lean very much on his expertise to try and work out what's going on within CLL cells. And there's a huge amount of investigation at Southampton trying to document these pathways. I haven't talked about all the other drugs. Someone mentioned Idella, and there are many other drugs being thrown at these pathways. You need to understand the pathway in order to make the drug more effective, more discriminatory, more useful. Andy, of course, is the drug czar, and he probably will be talking at the next meeting, and John Strefford you've heard from. Science is absolutely international, Brexit or not, it won't make any difference. And we work with people all over the world. I've just put a couple of our collaborators from Italy who we work with very closely. Chris Pepper, some of you know from Cardiff, has been part of this. Members of the European research in CLL, Eric, we're in constant touch with. We work very closely with many people in the United States. This is not a single effort. This is an international collaboration, all sciences. Otherwise it doesn't work. And we've had funding from these people. That should be Bloodwise, sorry about that. And this is our institution in Southampton. Thank you very much.